wanted to see if there was an optimum pH for the trypsin enzyme. And enzymes are going to be molecules that help catalyze chemical reactions, and they do that by lowering the activation energy required for that reaction. Um, and specifically, we were working with the enzyme trypsin in this lab, and that's an enzyme that breaks down proteins by breaking down the smaller peptide bonds and chains within that protein. And trypsin is known as a hydrolase molecule, which is a class of molecule or enzyme that uses water to break down chemical bonds. So in humans, trypsin is synthesized in the pancreas in an inactive form called trypsinogen. That pH condition is usually more acidic, around 2, pH of 2. That trypsinogen is then sent to the small intestine, which has a pH of around 8 or so, and it's converted into that active form of trypsin, which is going to be used to catalyze some of those reactions. And in bacteria found in nature, which has the active trypsin in it, found in it being synthesized naturally, they usually have a pH ranging from 7 to 10. So again, this group wanted to see, is there an optimum pH range for the trypsin enzyme? Is there going to be a pH where this enzyme works best? So we hypothesized that the optimum pH for the trypsin enzyme to work would be pH 9. And we did this because in nature, typically, you see it around that range. Again, that bacteria, it had a bigger range, 7 to 10. In humans, you're going to be regulating around a pH of 8 in that small intestine, which is where naturally that trypsin is used. So we decided we were going to test pHs 7, 8, 9, 10, and 11, just to clearly test the range to see if there was any activity in some of those extremes that we thought might not have much activity. So we really wanted to make sure our data would support that this pH 9 is clearly the better pH for trypsin. We also decided to use 4 replicates. That way we were catching outliers within each of those pHs we're testing, to measure the variability more accurately, and also to find a more accurate reading of the enzyme activity in each pH level. So we had the opportunity to prepare our solutions the day before. So in each of the centrifuge tubes, we put about 500 microliters of the azo casein and 450 microliters of the reaction buffer, and that differed between the pH 7, 8, 9, 10, and 11. So, and not only did we have 4 replicates testing each pH, we also decided that with this experiment we wanted negative controls as well. So we had another set of centrifuge tubes with pHs 7, 8, 9, 10, and 11 to test, to use as our negative control group. So again, we put the azo casein in each of the centrifuge tubes. We put the reaction buffer in each of the centrifuge tubes. And the negative controls ended up getting 50 microliters of distilled water, because that wouldn't have any effect on the reaction or whatever reaction might have occurred in those samples. And in the experimental group, we added 500 microliters of trypsin. But that was the next day once they were thawed. And so after the enzyme trypsin was added to our samples, we had to start a timer for the reaction, and we decided that we would keep our samples at room temperature. So each of them were submerged in a room temperature bath for 20 minutes at 37 degrees Celsius. After they had been in the dry bath for 20 minutes, you put on gloves to handle the TCA, which was going to be added to each sample. and we added 500 microliters of TCA to each of those samples. And so TCA is a solution that's going to break down reactions. So after the TCA is added, all of those centrifuge tubes, all of those samples were then centrifuged at 12,000 RPMs for 2 minutes, which then clearly made a pellet and the supernatant in each of the centrifuge tubes. So that pellet that we found in each centrifuge tube was the uncatalyzed particles in the reaction, and what was in the supernatant was actually going to be the catalyzed parts of the reaction. So after reading the absorbance, or the point for reading the absorbance was, if there was a higher absorbance, there's more catalyzed particles in the supernatant, so therefore there's more enzyme activity going on. So that's what we're trying to find out. And so again, that's what that centrifuging is going to do after adding the TCA. So that TCA is going to break down everything that was in that reaction, and then the centrifuge is going to clearly separate what was catalyzed and what was not. So centrifuging happened after making sure that the spectrophotometer was set to read 440 nanometers, and after blanking it with distilled water. So as you can see in our results, we had some funky things going on. So here, our experimental group is here in the blue, and our control group is here in the red. So what you can see is that there's not much of a difference between the absorbances you see in the experimental group versus the control group. There's actually about a 21% difference between those values. So it raised some red flags for us in the beginning. So you can see the negative controls actually have a high absorbance rate compared to the experimental group. We saw an average about 0.8 for the absorbance. In the experimental group, most were reading about 1.15. Clearly at pH 9, there was a 1.1 or 1.2 absorbance reading. So in a way, you can see that pH 9 did have better values, which correlates to pH 9 being the better pH level for the trypsin enzyme. And also the standard deviation between each of our experimental values was about 5%. So there wasn't much deviation actually between the values. So our t-test down here as well was not as helpful as we wanted it to be. But you can see this is for our control versus the experimental. It's less than 0.05, meaning that the enzyme did have an effect on the catalyzing of the reaction. However, the t-test compared with the experimental group shows that there might have been too much enzyme in each solution to clearly test if there was going to be an optimum. So also, we could have, with our negative control, one of our negative controls was the room temperature. We decided that it was going to be 37 degrees Celsius. However, we could have adjusted that based on the part group. They tested the temperature optimum for trypsin, and they found that the optimum range was going to be between 48 and 68 degrees Celsius. So if we would have increased our temperature, maybe we would have seen different results. So, and they had found that that temperature was optimum in bacteria that uses the trypsin enzyme. although our results weren't as promising or weren't what we expected or wanted, the Love group also tested pH, and their results clearly showed that there was an optimum between 7 and 9. So we were around that range. So some of that variability could have been too much enzyme in the way we were pipetting, and maybe pipetting in other areas of the experiment. Maybe some of our values and levels were different in each of the samples. So that would have been a huge issue. One thing we concluded after seeing that the absorbance rates were so high, or the readings were so high in the control group, we decided that maybe there was a malfunction in the spectrophotometer, and perhaps if we had tested with a different spectrophotometer, we would have had different values. However, the average for pH 9 was a little higher than each of the other pHs tested. So that was what we could conclude from our results, that pH 9 is the optimum pH for the trypsin enzyme.